Hello guys, welcome to my channel. In this video we can see detail about Agrobacterium tumefaciens, a pathogenic organism which cause ground gall disease to plants. Now let's see the general characteristics of Agrobacterium tumefaciens. It's gram-negative bacteria, rod-shaped bacilli, and it shows a swimming motility by 1 to 6 flagella on the one pole of bacteria, and its rise will be 1.5 to 3 into 0.6 to 0.1 μm, which is micrometer, and the capsule is absent. Also the endospore is also absent. Respiration, it leads to aerobic respiration, and the optimum temperature for growth is 28 degrees Celsius, and optimum pH will be 7 in neutral, and it's belong to the Rhizobiaceae family. And soil is a habitat of Agrobacterium tumefaciens. It was recently called as Rhizobium radiobacter. It is reclassified based on 16S RNA sequence. Its habitat of soil belongs to Alpha Proteobacterium, which means nitrogen fixing legume symbionts. Unlike others, it's tumor producing organism. Identified by J.N. Noel, Rhizobium radiobacter in 1982. As previously, Smith and Townsend described as Agrobacterium tumefaciens in 1907, and the incubation period of Rhizobium radiobacter is 2.5 to 4 hours. Now, the transmission of disease. Agrobacterium tumefaciens. It is the soil phytopathogen that naturally infects plants' own sites, causes ground gall disease through delivery of transferred DNA from bacterial into host plant cells through bacterial type 4 secretion system by transferring Ti plasmid. Now, its virulence factors. Factors which supports pathogen for causing disease. There are some factors which includes Ta plasmid, which is tumor-inducing plasmid, virulence genes, virulence proteins, and acetosringon induction. Now its pathogenicity. Agrobacterium tumefaciens causes ground gall disease in plants. This bacterium transfers a segment of its DNA known as tDNA, which is transferred DNA, into the plant cells. This transfer is facilitated by a tumor inducing plasmid carried by the bacterium. Once the tDNA is integrated into the plant genome, it induces the formation of galls, which are abnormal growths or tumors. The tDNA encodes genes that stimulated the production of plant hormones like auxins and cytokinins, leading to the uncontrolled cell division and tumor formation. These tumors provide a hospitable environment for Agrobacterium to thrive. And it is the mechanism which Agrobacterium tumefaciens enters its Ti plasmid into host cell and gets integrated into host cell genome in plants and its disease. Agrobacterium tumefaciens majorly cause ground gall disease in plants, which enters the plants through wounds in roots or stems and stimulate the plant's tissues to grow in disorganized way, producing swollen galls. Galls can interrupt the flows of nutrients and water within the tree, reducing overall plant growth and vigor. Young plants with many galls encircling the main stem are the most severely affected and can be killed by the disease. And its laboratory diagnosis. Gram staining shows gram-negative and rod-shaped bacilli. The motility test by hanging drop method shows motile, a swimming motility by 1 to 6 flagella on one pole of the bacteria. Colony morphology of Agrobacterium tumefaciens while culturing. They were used complex media such as lysogeny broth (LB media), alternatively known as Luria Bertani media or Luria broth, and yeast extract peptone media. White or off-white color, smooth, cream or opaque, round or irregular colonies produced. Colony morphologies of Agrobacterium tumefaciens, genomic species G1 and G4 are one name medium. A. Orange brown pigmentation of genomic species G1. B. Lavender pigmented and mucoid phenotype of genomic species G4. Also grow on 2% NaCl and ferric ammonium citrate medium. Biochemical tests for identify Agrobacterium tumefaciens. Catalyze test positive, oxidase test positive, indole test negative, MR which means methyl red test negative, August procarase positive, nitrate reduction positive, 3 keto lactose production positive, and citrate utilization source negative. 2% NaCl tolerant tests are used to identify Agrobacterium tumefaciens. And the confirmation test for Agrobacterium tumefaciens is antibiotic resistant test showed that all the isolated were resistant to rifampicin and tetracycline and susceptible to kanamycin and cefuroximide, which was supported by the Cohen et al. And Carthy et al. Treatment. The antibiotics which are used to treat Agrobacterium tumefaciens in plants are carbancillin, cefotaxamine, and vancomycins are antibiotics widely used to suppress Agrobacterium after genetic transformation. And the control measures are reduce plant wounding as much as possible. Source clean plant materials from responsible breeders and propagators. And these are the details about Agrobacterium tumefaciens.